Hello, everyone, and thank you for attending today's webinar entitled Quantitative Evaluation of Biomarkers for TGF-beta-Induced Epithelio-Mesenquimal Transition in Biochemical, Cellular, and 3D Spheroid Model Systems. We're excited to welcome our speakers today, Dr. Anthony Wood, Senior Director of Product Design and Strategy at Cell Signaling Technology, and Dr. Ernest Heimseth, Application Development Scientist here at Agilent Cell Analysis Division. Before we begin, I just want to cover a few housekeeping items. On your screen are multiple application engagement tools that you can use. These tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. Today's presentation will be followed by a live Q&A session, and we ask that if you have any questions during the webcast, please submit them through the chat box labeled Ask a Question, located on the lower left side of your screen. We will do our best to get to as many as possible, and the rest will be answered later via email. Please know that we do capture all questions. Additional help materials, including the application note and poster that today's presentation is based on, and information about the Agilent BioTek instrumentation used and cell signaling technology are available in the related content module. We encourage you to click on any links that you may find useful. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. Webinars are bandwidth intensive, so closing any unnecessary browser tabs will help conserve your bandwidth. For the best audio quality, please make sure that your computer speakers or headset are turned on and the volume is up so you can hear our presenters. Please note that an on-demand version of this webcast will be sent to you via email in the following days. Again, I want to thank you all for joining us today. And with that, I will turn things over to Tony.
Thank you, Bear, for the introduction. As Bear mentioned, my name is Anthony Wood from Cell Signaling Technology. And on behalf of my co-presenter, Dr. Ernest Heimseth from Agilent Technologies, we'd like to welcome you all to this webinar, and where we are describing methods to quantitatively evaluate biomarkers for TGF-beta-induced epithelial-mesenchymal transition in a variety of different models, ranging from biochemical through to 3D spheroid models. This webinar is really the outcome of a partnership between Agilent Technologies and Cell Signaling Technology. Agilent is a global leader in life science, diagnostics, and analytical laboratory technologies, while CST is dedicated to providing the world's highest quality, innovative research products to accelerate biological understanding, with specialization in recombinant monoclonal antibodies. Our companies have joined forces to demonstrate the power of combining cutting-edge cell analysis instrumentation and software with rigorously validated antibody reagents to generate informative insights into complex biological problems. Every antibody developed at CST is subjected to rigorous testing according to what we call the hallmarks of validation. This is a suite of testing strategies designed to ensure that every antibody is specific for its intended targets. Importantly, these hallmarks are applied independently across applications for which an antibody is tested. It's important to note that not all antibodies work in all applications, and they must be validated independently. Today's major theme of today's presentation is cell signaling, where CST has long had a position of innovation and leadership. One example of this is the Phosphocyte website shown here, where we provide curated, annotated information on post-translational modifications across the proteome. We've recently partnered with the Cantley Lab and developed a kinase predictor tool, which is now hosted on the Phosphocyte website, as shown in the bottom right. What I'm showing here in these screenshots are some visual representations of post-translational modifications of the SMAD2 protein, which is one of the topics of today's presentation. A central theme today is the TGF-beta signaling pathway, and specifically its role in the process of epithelial-mesenchymal transition. TGF-beta signaling is really a constellation of many different signaling pathways, over 30 ligands activating ligands activating multiple pathways. It plays very complex roles in development and disease, and it's difficult to ascribe a specific role to TGF-beta signaling. For example, it can be oncogenic in one context, or it can be tumor suppressive in another context. It's known to be a major driver of fibrotic diseases, while also playing an important role, particularly in the immune system, in suppressing immune cell function. Its importance in development and disease is underscored by the fact that there over 32 active clinical trials currently involving TGF-beta, with another 77 clinical trials currently recruiting. And these range from small molecules to vaccines, monoclonal antibodies, etc. So many different modalities, all looking to manipulate in some way the TGF-beta signaling pathway. As I mentioned, it plays a very important role in specific diseases. We know that TGF-beta signaling is a major driver of fibrotic diseases, such as idiopathic pulmonary fibrosis or non-alcoholic fatty liver disease. It's a central driving factor because many target genes are factors that actually contribute to matrix remodeling and can be fibrogenic. In addition, we know that TGF-beta signaling plays an important role in the tumor microenvironment through a number of mechanisms. For example, it can modulate the function of immune cells, generally having an immunosuppressive function on immune cells, while also it can help to remodel the tumor microenvironment by regulating the transcription of various remodeling factors such as matrix metalloproteases. For today's presentation, we're going to be focusing on TGF-beta signaling's role in the process of epithelial-mesenchymal transition. This is a cellular differentiation process whereby epithelial cells lose their epithelial character and become more mesenchymal-like in nature. They adopt a more migratory phenotype, and many of these features are consistent with those observed during cancer cell metastasis. And the TGF-beta pathway is a major driver of factors, including transcription factors, that are involved in the epithelial-mesenchymal transition. We offer a number of reagents and assay antibody collections that allow users to actually interrogate epithelial-mesenchymal transition at various levels. One such example is shown here. We actually have a second example where every reagent in this kit is designed to be used in an immunofluorescent or immunocytochemistry assay. And we feature a number of these antibodies in the results that we present today. We also present data for a target known as HMGA2. This is not included in those earlier collections, but it is one of the key transcription factors that allows TGF-beta to regulate the transcription of downstream target genes that induce EMT. And these are just some data shown here, an immunofluorescent image showing the substantial induction of HMGA2 expression following treatment with TGF-beta. And this is described in this paper listed here. And these data were generated actually during the development of an antibody targeting HMGA2. So when we talk about the TGF-beta signaling pathway, there really is a canonical pathway that many people are familiar with, and this relies upon the SMAD proteins to transduce the signal from the cell surface to the nucleus. There are two major pathways within this superfamily. There's the TGF-beta pathway shown on the left here, and then there's the BMP-driven pathway shown on the right. Both of these have cognate receptors, which then recruit SMAD proteins upon their activation. The TGF-beta family receptors recruit SMADs 2 or 3, while the BMP family receptors recruit SMADs 1, 5, or 9. These recruited SMAD proteins are activated and form a complex with a co-SMAD, SMAD4, and this complex translocates to the nucleus where it regulates the expression of target genes. There are a number of related pathways in this overall pathway diagram. Some of these other pathways you may be familiar with, they're generally thought to be involved in fine-tuning or modulating the pathway in response to TGF-beta signaling. In order to activate this TGF-beta pathway, it typically relies upon activation of the receptors, which then recruit the SMAD proteins and activate them through kinase-mediated phosphorylation. So there's a series of phosphorylation events that are indicative of activation of this pathway. In addition, as I mentioned, these protein complexes upon phosphorylation are translocated through the cytoplasm and ultimately end up in the nucleus where they activate or regulate the transcription of target genes. So primary readouts of SMAD pathway activation would include SMAD phosphorylation and SMAD translocation. These are actually among the most effective readouts of pathway activation. So if you're, for example, involved in therapeutic discovery and looking for a modulator of the TGF-beta, the canonical TGF-beta pathway, you would likely be looking for factors that modulate SMAD phosphorylation or SMAD translocation. Cell signaling offers a number of reagents to allow you to interrogate the TGF-beta SMAD pathway. If you're looking at of course, total proteins. We offer a number of target-specific antibodies. I've shown a few of them here. This is just a small selection of all that we offer. And as you'll note, that many of these antibodies are validated for different applications, and they're not all consistently validated for the same applications. That's because functionality in any given application does not predict the functionality of that antibody in a different application. So we have to independently verify and validate these antibodies in each tested application. So it's important when you're selecting reagents to select a reagent that has been validated and approved for its intended application. If you're looking to look at activation of a signaling pathway, you're probably looking for post-translational specific modifications. And CST has real leadership in this space. Here's some examples of antibodies against specific modification sites in SMAD proteins that are representative of activated SMAD proteins. Again, these are not all validated in all applications because every antibody needs to be independently tested, and functionality does not always predict across validation. I've highlighted here many antibodies that are approved for IF, which is a precursor to high cont analysis, which is part of the analysis that we do today. These are just some examples of some data from some of our phosphospecific antibodies in the TGF-beta SMAD pathway. As you can see on the bottom left, clone 138D4, it's approved for Western blot, but is not approved for IFIC. If you wanted to do an IFIC experiment, you would need to use our clone E8F3R because it has been validated for immunofluorescent immunocytochemistry. And an example on the right, an example of another clone that happens to work for chromatin immunoprecipitation, but not for the other applications. So it's very important when you're selecting reagents to understand how they're validated and for what they are actually approved for use. We also offer, of course, many antibodies against total SMAD proteins. And I mentioned our SMAD4 antibody here. This is actually a multifunctional antibody. It is validated for multiple applications. But because the SMAD4 is a co-SMAD that partners with SMADs 2 or 3 or SMADs 1, 5, or 9 and forms a complex that translocates to the nucleus, it actually serves as a nice orthogonal readout of activated signaling. So we use this in our experiments in this study to provide an orthogonal readout of pathway activation. We know that literature citations are very important to the scientific research community, and we do pay attention to this. I'm showing here some data from the organization CITAB, which tracks the citations of antibodies in the primary literature. And if you do a search for SMAD2 on the CITAB website, you will note that we far and away are the leading company cited for the SMAD2 antibodies in the primary literature. We also curate this data on our own, and so the graph on the right shows providers on the y-axis, showing how a search for SMAD2 reveals that cell signaling technology reagents are the most highly cited in the SMAD space. So we take this as pretty compelling evidence that our antibodies are widely used by the research community in their published research. We also, although not as well known, offer a number of reagents for the ELISA assays. And so I've highlighted some of them here, focusing on, again, the TGF-beta SMAD pathway. We have a number of ELISA kits and reagents that allow you to interrogate both activated SMAD pathway and the total proteins in the pathway. So these are approved for ELISA, and we indicate that with the icon or the chiclet indicated by the red arrow. We offer these ELISA kits in a number of different formats. We have traditional sandwich ELISA kits called PathScan ELISA kits. We also offer them in a rapid format, 2 different rapid formats, one known as the FastScan and one known as the PathScan RP, which stands for Rapid Protocol. And so if you're looking to interrogate the TGF-beta SMAD signaling pathway through ELISA, we have a number of options available for you, and we demonstrate their use in this webinar today. I'm also excited to announce that this year we're offering a new product line, which is matched antibody pairs. So these are ELISA pairs that have been pre-validated for use as a pair in ELISA-like assays, but they're sold in a carrier-free format and not as part of a complete kit. So they can be then adopted for use in a variety of different platforms, trying to make that as flexible as possible. So that's a new launch, and we're constantly adding new pairs to our portfolio. And if you have any questions about our ELISA portfolio, we do have a resource center that you can find at cellsignal.com. Don't hesitate to visit if you have questions, or of course, reach out to CST at any time. So now I'm going to turn the mic over to Ernest, and he's going to walk you through the data we generated. So the objective today was to demonstrate high throughput measurement methods for measuring TGF-beta induced epithelial mesenchymal transition in multiple models. We look at this at a biochemical level using ELISA assays from cell signaling technology. We also look at it in a 2D cellular model system using fluorescence wide field microscopy. And we also look at that in 3D cellular spheroid models using fluorescence confocal microscopy. All of these data were generated using recombinant monoclonal antibodies from CST on a single platform, the Cytation C10 confocal imaging reader. Over to you, Ernest.
Thank you, Tony. First, I'd like to give a brief introduction to the instrument that we're carrying out today. As Tony had mentioned, this is the Agilent BioTek Cytation C10 confocal imager reader, which is a versatile instrument capable of conventional microplate reading using a quad monochromator and PMT-based sensor. In addition, it has 3 different modes of light microscopy imaging: transmitted light, which is phase and bright field microscopy, LED-based wide field microscopy, and laser-based spinning disk confocal microscopy. In addition to the hardware features of the C10 is controlled by our software, Gen 5, which is a versatile platform that can take you from image acquisition to figure presentation. It's also 21 CFR Part 11 compliant as well, which is conducive with GXP laboratory requirements. So starting off with the biochemical scale, we wanted to quantify TGF-beta1 induced MAD phosphorylation by means of an ELISA. To accomplish this, we used 2 different ELISA kits by cell signaling. The first measures phospho-SMAD23, while the second measures total SMAD23. These are colorimetric assays, so absorbance at 450 nanometers is our readout signal, which can be measured using the plate reader capabilities of the Cytation C10. And here I'm just illustrating an experimental workflow that was used. And again, conceptually, this is what we're looking at upon stimulation of TGF-beta and its receptor. SMAD2 gets phosphorylated. And so what we're doing is assaying both the phosphorylated state of SMAD2 as well as total SMAD2. First thing we did is we seeded cells in a 96-well plate, underwent a serum starvation period, as well as TGF-beta treatment, and then harvested cell lysates. We applied those cell lysates to the ELISA kits, and then developed the assay as per instructions. We then took those and read absorbance 450 nanometers using the quad monochromator. And then from there, we can build out a dose response curve for TGF-beta1 and SMAD23. And these were using, the cell line that we were using is A549 cell line. So the Gen 5 software has a built-in computational functionality. So here, what you're looking at on the left is just a plate view of what the values you would get from an absorbance read. And on the right are different calculations that are possible in Gen 5 to take this raw data and convert it into meaningful information. So in the case of measuring SMAD23 phosphorylation, Gen 5 allows us to easily derive dose response curves. So these black points show the correlation of SMAD23 phosphorylation with increasing concentrations of TGF-beta as represented by this fold change of no treatment. And a sigmoidal curve, 4-parameter curve can be fitted to derive an EC50 for these experimental conditions. Because these ELISAs targeting both we've assayed both phospho-SMAD23 and total SMAD2, we can evaluate and normalize for any relative changes in total SMAD levels, which are shown in blue. If you're familiar with quantitative Western blotting, this is like equivalent to accounting for total protein. And these values in the black are actually have been accounted for based on the values of At the next level, imaging-based cellular assays can complement ELISAs by providing quantitative spatial information that visualize phospho-SMAD nuclear translocation, as shown conceptually here on the left, where we want to see not only does SMAD2/3 get phosphorylated, but where is it spatially localized in these cells? And here's also another conceptual view of the workflow is we're going to take this assay, immunostain with a phospho-specific antibody, image them using white fluorescence, and then get quantitative values. Before I get into the data, I'd like to first introduce one of the newest features of the Cytation C10, which is automated water immersion imaging. So water immersion imaging is accomplished using microfluidics, which delivers deionized water to an adapter cuff fitted on the objective. This design of automated water immersion retains the high throughput capabilities of the Cytation C10. But to appreciate the importance of water immersion imaging, I'd like to first discuss the concept of numerical aperture, which describes the light collecting capabilities of an objective. So if you have an object here, say the spheroid or any kind of biological sample, that sample will emit some kind of fluorescence if it's a fluorescence-based assay. And the amount of signal you'll collect is a function of the numerical aperture, which depends upon the half aperture angle shown here in alpha. So the larger the half aperture angle, the more light and the more signal you're going to collect from your sample. So as one example of how this works is, here is images of the same 1 micron wide fluorescent bead imaged with objectives of different numerical apertures with the same exposure settings. As you can see, the objective with the higher numerical aperture allows for more signal to be collected relative to the lower numerical aperture. This is quantified here below in this line scan shown here. As another example, here are some fixed and stain adherent cells that were imaged using a 60x objective with an air objective or a water immersion objective, again, with the same exposure settings. Using water immersion imaging to collect more signal while minimizing exposure has 3 major advantages shown here. It will ultimately be much gentler on your sample. It would allow you to collect and quantify more signal, which is especially important with dim signal. And lastly, it would dramatically reduce optical distortions when imaging aqueous samples, such as if these cells are in PBS. With that being said, wide-field water immersion imaging was used at the cellular level to quantify SMAD phosphorylation and nuclear translocation in adherent A549 cells. This is an endpoint assay which allows us to immunostain with a phospho-specific antibody listed here. And here are some of the other reagents we use, our cell lines. And again, we're using the C10 in wide field mode using phospho-SMAD2/3 as our readout. So as we see in control cells, there's little phospho-SMAD2/3 detected with these acquisition settings. However, in the presence or when treated with TGF-beta, we see a robust signal as shown in red here. This is red, and this is nuclear signal. Additionally, we can attribute this signal to TGF-beta signaling because cells pre-treated with a TGF-beta receptor inhibitor, SB431542, in this samples that response is gone. Oh, sorry, let me just, it's also worth pointing out that this is phospho-SMAD2/3 is a relatively low abundant signal, so which is another reason why we wanted to use water immersion to kind of extract as much signal as we possibly could. Gen 5 allows us to quantify phospho-SMAD2/3 fluorescence by segmenting the nuclei, as shown here, using the DAPI channel. I'm not showing the DAPI channel, but using that, we can segment and identify each individual nuclei from these cells. And then quantify SMAD2/3 signal within each individual nuclei. This allows us to quantify this response. And just pointing ahead, this same kind of image analysis is the same that's going to be done in subsequent experiments, both at the adherent 2D cell level and as the last biological 3D level. So in addition to SMAD2, SMAD2/3 phosphorylation, we can quantify markers of EMTs in the same analysis workflow as shown here. One being HMGA2, ZEB1, and SLUG. So we see both in the absence and the presence of TGF-beta, and that quantified signal. So when with the high throughput capabilities of the Cytation C10, along with the water immersion imaging capability, we can expand our assay beyond single point concentrations in samples and instead build out a dose response curve of multiple replicants, as I'm illustrating here, that multiple wells you can treat with different concentrations of TGF-beta along with multiple replicants. When imaging in multiple XY positions, such as multiple wells, and it's important to maintain focus from point to point. For these high throughput imaging assays, laser autofocus was used. This technology triangulates the set focal point by reflecting an infrared laser off the bottom of the well and adjusting the Z height as needed from one well to the next. And as mentioned before, the microfluidics cuff of the C10's water immersion system enables that the bolus of water is maintained even when pivoting from one well to the next. So using the high throughput imaging capabilities of the Cytation C10, this allows us to build dose response curves. This allows us to build dose response curves for TGF-beta stimulated nuclear phospho-SMAD2/3, as well as the EMT markers HMGA2, ZEB1, and SLUG, as shown on the left graph. In addition, dose response curves showing the efficacy of the TGF-beta1 receptor inhibitor, SB431542, were generated as well, as shown on the right. At the largest biological scale, we employed a multicellular spheroid model to evaluate TGF-beta1 induced SMAD signaling. In this assay, A549 spheroids were treated with TGF-beta1, then fixed and immunostained with the same phospho-2 antibody used in the 2D adherent cultures that I just discussed. Once these sample preparations were complete, these spheroids were then positioned at the bottom of a 96-well plate and imaged using C10's confocal mode. There are a few concepts of resolving confocal microscopy and how these samples were imaged that I'd like to introduce as well. So as I mentioned, the Cytation C10 uses a spinning disk confocal technology, which provides distinct advantages over single point scanning systems by allowing users to decrease excitation energy to illuminate their sample and reduce phototoxicity and photobleaching. The Cytation C10 can house two different spinning disks, which can be selected and put into position using the Gen 5 software. Now, confocal gets its name because a pinhole is placed in a conjugate focal plane of the sample. And by doing so, autofocus light is emanating from the sample is dramatically reduced, as illustrated with these 4-micron beads imaged in both wide field and confocal mode. Another enhancement of the C10 is the addition of a 60-micron deep section disc, which reduces background in thick biological samples, allowing more meaningful, detailed data to be collected. There are many sources of background. However, the specific background that the deep sectioning disc address is called pinhole crosstalk, which I'll briefly walk through next. Now, confocal gets its name, as I just mentioned, because you have a pinhole or an array of pinholes in a conjugate focal plane. So when you excite any given focal plane, you want that pinhole to be in a conjugate focal plane on the other side of your objective. And so in a spinning disk situation, I'm just illustrating one pinhole, but you're going to have an array of pinholes. However, the excitation light doesn't just excite fluorophores wherever you're in focus, but it excites fluorophores throughout your entire sample, if it's a big sample. Now, this causes a problem. is that because those out of focus fluorophores are excited, they're going to be out of line with the conjugate plane, which your disc or your pinholes are set up. What this does is this causes that signal to bleed into adjacent pinholes in your array, causing your sample to show up as kind of background haze, as shown in this spheroid. So what we did is we developed a disc called the deep sectioning disc, which actually has the same array of pinholes, but they're fewer and more spaced out. That way, in any given focal plane, you will eliminate much of that pinhole crosstalk, the background due to that pinhole crosstalk. So because A549 spheroids is susceptible to the same pinhole crosstalk, they were imaged using the deep sectioning disc in order to achieve the most refined image possible, as shown here. As you can see, TGF-beta treatment elicited robust phospho-SMAD2/3 and nuclear translocation response in A549 spheroids, shown on the right image, just as they did in those adherent cultures. Again, the high throughput capability of the C10 allows for multiple replicates across various concentrations of TGF-beta1 to be imaged in an automated fashion. And this allows, and because of the downstream image processing and analytical capabilities of the Gen 5 software, we can fold into those capabilities into the workflow. Dose response curves from these spheroids can be generated. So to quantitatively evaluate the influence of TGF-beta in a more dynamic context, two assays were set up. In the first, those preformed A549 spheroids were fully embedded in a collagen matrix and exposed to various concentrations of TGF-beta1. And the correlation, they were allowed to culture for about 4 days. And so what we saw is a correlation between TGF-beta1 concentration and overall spheroid size, as quantified here on the right. In a variation of this assay, the spheroids can then be laid on top of a collagen matrix, which allows for individual cells to migrate away from the core spheroid, which in doing so, which does so in a TGF-beta dependent manner, quantified here on the right. So not only do they migrate away from the core spheroid, but TGF-beta stimulated them to travel further, as quantified here on the left. So what we did, again, is using the same analytical workflow where we're identifying cells based on nuclear counts and tracking their distance from the core spheroid. And so this histogram on the right shows that in control cells, there still are some that migrated away from the core spheroid, but in the presence of TGF-beta, more cells traveled further away from the core spheroid. So we can verify we also verify two things from this kind of assay. So if we zoom into this region right here, the first thing we can verify is that TGF-beta1 is stimulating SMAD signaling as visualized and quantified by SMAD4 nuclear translocation, shown here on the right, left images, and on the right quantified here. Now, the second thing is that this outward migration is due to increased motility, typical of EMT, and not by increased proliferation. So in separate assays, we treated A549 adherent cells and tracked their motility, quantified here, and also we quantify proliferation rates seen on the right. Now, this is well documented that TGF-beta1 has an inhibitory effect on proliferation. So what I've shown you today is we showed that we can, using the superior reagents from cell signaling along with the imaging capabilities of the Cytation C10, we can assay SMAD signaling and EMT in 3 different scales: at the biochemical scale, using an ELISA kit in the multimodal plate reader mode. At the cellular level, using a phosphospecific antibody and wide field imaging coupled with water immersion imaging. And also in 3D multicellular context, using A549 spheroids and the same phospho antibody, SMAD2 and SMAD4 antibody, in addition to the confocal mode of C10, coupled with the deep sectioning disc. So thank you very much for joining us. And with that, I'll be happy to take any questions.
All right, thanks so much, Tony. And as Ernest said, we can address some of the questions that came in. If you haven't had a chance to do so, please submit your question right now using the Ask a Question module on the lower left-hand of your screen, and we'll get to as many as we have time for. All right, so we have one here. Jason, do you want to take over?
Sure. We do have a couple questions that have come in. First one for CST. Are all antibodies developed by CST automatically tested in every application?
No, we actually put significant effort towards deciding what is the most likely applications for which an antibody needs to be used. And so we prioritize specific applications for specific antibodies in our testing. But if we do get requests, and we will then reevaluate an antibody that may not be validated for an application in that given application if there are requests for that.
Very good. And one for Agilent. Can water immersion be used in both wide field and confocal mode?
Yes, yeah, so the two are not coupled together. As I showed that we coupled water immersion with wide field imaging for the 2D adherent A549 cultures. Signal is fairly dim there, so I really needed it to bring out some dim signal in some of the samples. Certainly the same can be done with confocal. I didn't do it there because I found that the signal, it became a judgment call. I found that the signal was robust, and it was robust enough for those kinds of samples. One of the things to caution about is, especially with 3D imaging and the size, is with a water immersion objective, the working distance, the distance between the objective and the bottom of wherever your sample is much shorter. It's a shorter working distance objective. So that really limits how deep into a sample you go. So if you have a sample such as a spheroid or organoid or a tissue that spans a few hundred microns, the working distance of a water immersion objective is generally around 200 microns. So I think there can be limitations there depending on your sample, how deep into a sample you can go with a short working distance, water immersion or not. But I found that it was sufficient. I got enough sufficient signal for those particular samples that I didn't need water immersion. But still, there's nothing from stopping you from using it in confocal mode.
Okay.
As well as combining it with a deep sectioning disc.
Okay. And one for CST. Does CST make antibodies on demand?
Great question. So we don't have a custom fee-for-service antibody development business, but we do work with people in the field. If we get requests for antibodies come in, we discuss with someone, and we can even do it in collaboration with them if we decide that a campaign is definitely worth pursuing. So that's something that don't hesitate to reach out if you were interested. We've had numerous collaborations with people in the field working on specific reagents that meet the needs.
Okay, thanks, Tony. And Agilent, can spheroids be cultured long-term in the Citation C10?
Yes, absolutely. So in a long-term live assay, the C10 is capable with most environmental controls, except for humidity control. So generally, the way we do this here is in environmental controls, including like gas and temperature. So you can put them in there. The way I generally set them up is coupled to our BioSpa 8, which is a culture system with a robotic arm that will transfer your plate from where it's housed in the BioSpa 8 to the Cytation 10 and then image. And then once imaging is complete, it'll return it back to the BioSpa for long-term cell culture. So I've done this for development of things like spheroids, where if we want to monitor the development or outgrowth of these over a long period of time, say a few days or weeks, we can set up a protocol for it to image with whatever imaging interval, and then put it down. So it's kind of a standalone. Once you set it up, standalone, and you can come back, monitor it if you want, but then it kind of just runs on its own. So long-term culture is possible with the Cytation C10. Recommended to use the BioSpa to carry that for long-term culture.
Great, thanks, Ernest. And Tony, CST, how can you tell if an antibody has been tested but failed for a given application?
Great question. It's something that it's a question that we, again, do get frequently. The best way is to actually reach out to our tech support team, because many antibodies you will see are not approved for applications have been tested, but they haven't approved. But it's also possible they may not have been tested. So it's really best to reach out if you have a specific use case. And we are looking for ways in the future to better convey that information, because it is a commonly asked question.
Great, very good. And Agilent, what culture vessel formats are compatible with water immersion imaging?
Oh yeah. So we validated a variety of different formats, largely multi microplate formats, 96 well 34, 24 well glass bottom dishes like MatTek dishes, and a variety of different ones like slides, your general like histological type of samples. One thing that when it comes to plates is generally when you have an objective, especially a short working objective, the closer you get to the edge can cause problems. If the bottom of the wells has a higher, is more elevated from like the edge of the plates, you could physically run into those or bump into the edge of the plate. And so we generally have safety measures in mind in the software to prevent you from doing that, such as like the peripheral wells. So from there, it's generally, if you really want to get all 96 wells, search for a plate that has a very low well bottom that's almost flush with the casing of the plate itself. Besides that, I think most plate types or at least we've validated some of the most common types of plate formats and seem to be conducive with that.
Great. And here's another one, I think, for you as well, Ernest. In 3D assays, do you only have one organoid per well?
Oh, yes, that's correct. So the way that I set this assay up is I seeded one spheroid per well. And that was to give me, I guess, the most data resolution. So in one sample, in one well, I have one organoid, and I could just analyze that one. There's nothing stopping you if, a common way to do this is to seed individual cells, culture them until they expand into multiple spheroids. You can certainly do that and image multiple objects, multiple samples per well. We also have another feature, because I did it in one sample, one feature of the Gen 5 software is to create a beacon or an ROI. That allows you to save the coordinates of any given object in a well. So if you have multiple wells in a sample, you can set beacons for each of those objects, image them all, and then move on to the next well too. So the way I did it is I just seeded them individual spheroids per well. It's a little bit tedious, but if you are going to instead do your cultures from the start up, as I described, there's nothing from stopping you. You could totally do that, and there's ways to image that way as well.
Okay. And a follow-up question from another attendee, Ernest. How big is a spheroid in a well?
Yeah, so these spheroids were about, oh wow, that's about 300 microns diameter. So assuming it's a sphere, it'd be 300 microns deep. So that's about how big you can go. You know, the well is much larger than that. You run into issues of just the fundamental optics of imaging and confocal, of how deep you can go and start to lose signal. Your fluorescent signal starts to degrade. But so I targeted no larger than about 300 micron spheroid for these kinds of assays.
Okay, very good. And one last question. How large of a thick sample can be imaged with a deep sectioning disc?
Oh yeah, so that kind of ties into there too. So the idea is that the deep sectioning disc will eliminate signal coming from other planes, but there is limitations, because if the sample is too thick, it is beyond a certain thickness, you're going to start to reintroduce that out of focus light just the same. So by the spacing gives you some buffer to do that, and I would say the range the range you'll see the benefit is between 150 and 500 microns or thick into a sample. Beyond that, you'll start to reintroduce that pinhole crosstalk out of focus light. It's still going to be you're still eliminating a lot more background than you were with the regular disc, but that's where you'll start to see some of that background reintroduced. So I would say that the deep sectioning gives you a range, maybe maxing out at about 500 microns thick of a sample. And we're talking about like from the flush, from the bottom of the well or your imaging surface into the sample.
Okay, very good. And Bear, that looks to have been our last question.
All right. Well, thank you, Jason, for moderating, and I want to thank both Tony and Ernest for a great presentation. On behalf of Agilent Technologies, thanks to everyone that joined us today. If we didn't get to your question, we'll follow up you via email. And if you would like more information on cell signaling technology, please visit www.cellsignal.com. And to learn more about Agilent BioTek's products and services, including our archive of previous webinars, please visit us at www.agilent.com. We hope that you enjoyed today's presentation and have a great rest of your day.